Thank you for being with us today. I am Joanne DeGennaro, president of the Center for Excellence in Education. In 1983, I assisted the late Admiral H.G. Rickover, father of the nuclear-powered Navy, to introduce an exciting U.S. educational organization. The center is the home of the Research Science Institute, the first research program for high school students to provide STEM opportunities for top academic achievers. Now a program for 100 U.S. and international students. The Institute at MIT for 6 weeks each summer has nurtured over 3,000 students at no cost to them. In 2003, the center inaugurated the USA Biology Olympiad, an online competition for nearly 10,000 students each year to identify finalists to compete for the 4-member US team to demonstrate their talent at the International Olympiad. And compete the US team does. Each team member has medaled gold, silver, or bronze since the first US team participated in the IBO. This month, Team USA brought home 4 gold medals. The center is proud that its 2 newest programs, the Teacher Enrichment Program and the STEM Lyceum for Students, have reached thousands with online and in-person programs focused on underrepresented populations and Title I schools to pique interest in STEM studies and careers. Webinars, panels, newsletters, activities, and other information is provided. Thousands of students and teachers have participated in this rapidly growing program opportunities throughout the U.S. The center is governed by an active board of trustees, and programming is managed by a dedicated and brilliant staff with the help of program alumni, parents of alumni, volunteers, and of course, corporations. Since our organization's beginning, CEE has been financially supported by donations from individuals, corporations, organizations, and the government. CEE surely invites your consideration in your philanthropy to gift CEE. Please, if you should have time, review our website at www.cee.org, email, text, or telephone, or just drop in to say hello at our McLean office. The center is honored to sponsor this session as advisor to the Congressional STEM Educational Caucus. As advisor, a focus of today's conversation regards CRISPR. It is presented for members of Congress and their staffs to help them augment best policy and decision-making for education and for the public's better understanding of this scientific breakthrough. Before I introduce our honored guests and moderator, we are so honored and privileged today to have the co-chairman of the advisory speak to us, Senator Jake Auchincloss. He's serving his 2nd term representing the Massachusetts 40th. In addition to his work on the Committee for Transportation and Infrastructure and the Select Committee on Strategic Competition between the U.S. and China, his areas of focus include healthcare, clean energy, gun violence, and building a strong middle class. Jake was born and raised in Newton, Massachusetts, the son of a surgeon and a scientist. After graduating from Harvard College, Jake joined the Marines. He commanded infantry in Afghanistan and special operations in Panama. He has degrees in economics and finance from Harvard University and MIT Sloan. Today, he lives in Newton with his wife, Michelle, and their children, Teddy, Grace, and Audrey. And most important, along with their Labrador retriever, Donut. Congressman, the floor is yours, and thank you for your remarks this morning.
I appreciate the gracious introduction, and it's a pleasure to be able to join you. I apologize for the brevity and the circumstances on an iPhone. I'm currently at the middle of a hearing on drug prices here on the Capitol. I am the congressman from the Massachusetts 4th, and I'm in my second term, and I represent really the suburbs of Boston all the way through the border of Rhode Island. And I'm also the co-chair of the Congressional STEM Education Caucus alongside my Republican colleague, Neil Dunn. And I want to thank CEE for organizing this. And I also want to thank David Chang and Feng Zhang for joining. I'll note that they're both fellow Bay Staters, and we're very proud of the scientific and biomedical talent that Massachusetts is host to. The foundation layer for innovation in biomedical domains, in energy, in engineering, in cryptography and quantum, and so many of the exciting domains that policymakers are engaged in and that could change standards of living for consumers ultimately are predicated on high quality mathematics education. And this is really my passion when it comes to STEM education policy, is how do we ensure that young students in particular are getting the curriculum and the pedagogy around numeracy and mathematics that they deserve, so that they can, when they get past 8th grade, 9th grade, really accelerate into algebra, calculus, and more applied mathematics purposes. But it all starts with early numeracy. And indeed, some studies have shown that early numeracy at the K-3 level is in fact even more predictive of later literacy than early literacy is, because early numeracy helps cohere the executive function of young people's brains, and it helps them think symbolically as opposed to emotionally about challenging subjects. So it's also the basic foundation for critical thinking and reasoning as well, which is, of course, something that we prize in today's civil society. So we really need to have, post-COVID, a whole of government, whole of society effort to improve basic mathematics education. And that starts with telling the truth to ourselves about the fact that mathematics scores have dropped amongst Americans. American 15-year-olds, according to the latest program of international student assessment, have declined over the last 5 years in their mathematics proficiency. And that's compared to improvements for students in other nations. So when my colleagues on the Select Committee on China talk about outcompeting China through industrial policy and quantum or biotech or military applications, my response is always, stop worrying about these downstream domains and instead ask yourselves, do we have a stellar STEM education pipeline? Because if we are educating the world's best STEM talent, we're going to win. We're going to win in the economic and technological competition against our adversaries. But if we're not, we have impaired ourselves, perhaps grievously. So I'm absolutely committed to high quality STEM education. I think that really starts with superb early curriculum and pedagogy for math and numeracy. And I look forward to working with CEE and with Mr. Dunn on efforts to make that federal policy. Thank you.
Thank you so much. Very cogent remarks. And I appreciate that you took time from your hearing to present these remarks. Thank you again. Dr. Feng Zhang is a core institute member of the Broad Institute of MIT and Harvard, as well as an investigator at the McGovern Institute for Brain Research at MIT. co-director of the K. Lisa Yang and Hock E. Center for Molecular Therapeutics at MIT, the James and Patricia Poitras Professor of Neuroscience at MIT, and a professor of at MIT with joint appointments in the departments of brain and cognitive sciences and biological engineering. Zhang is also an investigator at the Howard Hughes Medical Institute. He is a molecular biologist seeking to improve human health by discovering approaches to modulate cellular programs, including returning diseased, stressed, or aged cells to a more healthful state. He is a recipient of many awards, including the Lemonson MIT Prize, the Tang Prize, the Canada Gardner International Award, and many others. He is a member of the National Academy, the American Academy of Arts and Sciences. He received his AB in chemistry and physics from Harvard College and his PhD in chemistry from Stanford University. He is also, and we are so proud, he is a 1999 alum of CEE's Research Science Institute, CEE Trustee. Our moderator today is David Cheng. He is also an alum of the Center's Research Science Institute, like Feng. He brings over 15 years of experience in technology and computer science to Arbor, a firm in Massachusetts, where he co-developed the Arbor Computational Discovery Platform. And he's a leading contributor to the discovery of Arbor's CRISPR nucleases and CRISPR-associated transposases. From 1919 through April of 21, David led Arbor as CEO. Prior to Arbor, he developed market-leading quantitative automating trading strategies at JP Morgan and Goldman Sachs. He's an inventor of autocomplete predictions. He's a member of the four-person team that represented the US at the International Biology Olympiad, the IOI. David received his master's in engineering and his BS in computer science from MIT. With that, the distinguished moderator and panelist will help us in better understanding the importance of CRISPR for citizenry. Feng, I turn it over to you and David. Thank you.
Thank you so much, Mrs. D, for the intro, and to Representative Auchincloss, for the remarks. I think we start off with just the basics. When it comes to kind of buzzy topics like gene editing, it's possible to let our imaginations get ahead of the current state of the art. So just to start, Feng, can you tell us, well, what is CRISPR?
Sure. Well, first of all, thank you, Joanne, for the opportunity to be here, and also thanks to Congressman Auchincloss for introducing this topic, and also thank you, Dave, for hosting me in this discussion. So CRISPR is a gene editing technology. So over the past 20 years or so, the human genome has been sequenced, and so now we know pretty much all of the letters in the human genome. And there are 3 billion of these letters. And the cost for sequencing DNA has also dropped, exponentially. So now it can cost less than, $1,000, maybe even close to $100 to sequence the full genome of an individual. So there's a lot of data being collected, both from healthy individuals and also from people who are affected by different types of diseases. By comparing the genomes of these individuals, scientists have discovered many genetic differences. So these sometimes are single letter differences, or these can be differences across multiple regions in the genome. These differences contribute to people's risk for disease. And for a small number of these mutations, they can directly cause disease. And so these are called monogenic genetic diseases. In fact, to date, there are about 5,000 of them identified. And so the tantalizing idea is then, what if we can go into the genome and be able to change those letters? If we know what is problematic, what about just going in and fix it? So this is where gene editing comes in. CRISPR is the latest sort of generation of technology that enable people to go into the DNA of a cell and be able to make these types of changes. And so it's been very exciting. A lot of researchers and a lot of drug developers are using it now to be able to treat disease.
And it's just the latest gene editing technology. Can you just kind of describe what is the big advantage of CRISPR over the other or previous generations of gene editing?
The genome is large. So the human genome has 3 billion letters, and there are 2 copies of the genome in every cell, so 6 billion letters. If I tell you that there is a specific letter that is incorrect, you will need a search system to go through the 6 billion letters to find that exact letter that is wrong. And then once you locate it, you can go and change it. So the development of the ability to edit the genome really depends on having a robust and rapid way to be able to find these mistakes within the genome. And so CRISPR is the latest generation of technology that enables very simple and also efficient identification of these mutations. But there are multiple technologies that came before, things called meganuclease or zinc finger nuclease, or transcription activator like effector nuclease, or TALEN for short. All these technologies that came before CRISPR are quite cumbersome and difficult to engineer. Basically, you have to build a brand new protein to be able to go find that mistake in the genome. CRISPR, on the other hand, is much, much simpler. You don't have to engineer the protein anymore. All you have to do is to give it a piece of RNA. And this RNA can be easily and inexpensively chemically synthesized to be able to find, to match the sequence in the genome and to be able to find this small mistake. And so to draw an analogy, the technologies that came before CRISPR is like having to build hardware. So for every app on your phone, imagine you have to build a new phone to carry out the function of that app. CRISPR, on the other hand, is like a platform where you have a single phone, but you can load all sorts of software on it to go after each one of these mutations in the genome. And those are in the form of RNA. So you have the CRISPR protein, which is called Cas9, and you can give it a specific RNA for finding sickle cell mutation, or finding Huntington's mutation, or many other of these genetic diseases. So that dramatic simplification in the ease of use is what really sort of helps CRISPR stand out.
Great. And you mentioned a few diseases and monogenic diseases and therapeutic applications. We'll talk about that more in a minute, but maybe before we get to that, can you describe the impact that CRISPR has had on basic research? I just had a quick look on Google Scholar, and these days it looks like there are about 75,000 to more than 90,000 scientific publications each year that mention CRISPR. So what are they doing with it? What kind of impact is it having at the basic science level?
CRISPR is being used very broadly as a research tool to help scientists understand how cells, how organisms, and also how diseases function. The way that scientists go about understanding biology is that usually they try to go and change something and see how that change affected the function of a cell or the health of a whole animal. And so in the human genome, for example, there are 20,000 genes. And different subsets of these genes are turned on in different types of cells to make it a skin cell or a liver cell or a brain cell. And so the function of most of these genes are not fully understood. And so what scientists might do is that they can use CRISPR to go and edit one of these 20,000 genes and ask the question, what happens if we inactivate this gene, or if we turn this gene on, or if we slightly change how this gene sort of works in the cell. So CRISPR, because of the simplicity, has made the change of things in the genome dramatically faster and also easier. And so researchers basically around the world now are using CRISPR as a way to perturb cells or animals and then be able to study their function. So just to give you one example of the dramatic sort of impact that CRISPR has had on research, one of the things that researchers do is that they use animals to model disease. So they use a mouse and they change something in the mouse to try to model different types of human conditions. Before CRISPR, it would take maybe a year or sometimes more than a year to be able to make one mouse model to study the function of a single gene. Nowadays, with CRISPR, you can make that same mouse model in a matter of weeks. So within 3 weeks, you can get a mouse model, and then you can also do this at a much larger scale. So you can make dozens of models at once. And so if you put this in the context of, say, a graduate student's tenure, a PhD student usually spends somewhere between 5 to 6 years in graduate school to complete their PhD. If it takes a year to 2 years to make a mouse model so that the student can carry out their project, that is a third of the time that they have in graduate school. And so things would move a lot slower. But if they use CRISPR to make these mouse models, they can progress with their projects much, much faster. And that is one of the powers of CRISPR. And so this type of acceleration has really permeated all different fields of biology. And that's why you see so much mentioning of it within the literature.
Great. Now, yeah, let's talk about therapeutics specifically. So what does gene editing enable that was previously not possible, say with small molecules and other biologics like antibody therapies?
So what CRISPR has really enabled today is that because of genome sequencing, we're identifying underlying genetic causes for disease. And so what that means is that we know exactly what is mutated in the genome. And if we can restore that mutation back to the wild type, the conventional letter in people's genome, then we can reverse the disease pathology. So this is something that is very difficult and sometimes impossible to do using small molecules or using other forms of therapy like antibodies or other protein-based therapeutics. So gene editing allows us to go into the cells and be able to directly make those changes. So this is very powerful because if you can change the genome, then you fully restore the function of the cell. And you only have to do that once. So once you fix the the mutation, then the cell has been changed and you don't have to change the cell again. So CRISPR and gene editing based therapeutics are one-time cures for disease, whereas small molecules or even protein-based antibody or other therapeutics, you have to keep on taking the drug. And so the patient maybe have to take it every day, maybe every week or so. And so this type of single treatment cure can really dramatically change patients' lives, and it makes compliance much, much easier.
So I can see that working for monogenic diseases where there's a clear genetic driver, of a disease and, really getting to the root cause and editing that pathogenic mutation to provide a functional cure. What about some diseases that have been, very intractable, like neurodegenerative diseases, like ALS, where there are some familial forms, but there's also many sporadic cases of ALS. Is there, some approach for gene editing to move the needle for some of those difficult indications as well?
There are several ways that people are now using CRISPR to both understand and also treat a disease. So for something where the disease is very complicated, like neurodegeneration, one of the reasons why we haven't been able to develop very good therapies for them is that we don't fully understand the underlying. Using CRISPR, researchers are now able to take all the things that we learn from humans and be able to generate as realistic as possible, cellular and also animal models to better understand the underlying cause and mechanism of those disease. And by understanding the disease more, we're going to be able to develop better and more accurate therapeutics to be effective. And at the same time, there's also promising approaches for using gene editing to directly treat these complicated diseases. These diseases are usually not caused by a single genetic mutation. That's why they're called complicated or complex diseases. So rather than going and changing specific mutations, the way that CRISPR might be used is that people use CRISPR to find out what are some of the master regulators in the cells that underlie these diseases. And maybe we can use CRISPR to be able to modulate, either turn it on or shut it down in the cell, and to be able to use the master regulator to change the outcome of these diseases. And that is quite promising as well.
Yeah, talking about, from promise to reality, it's really exciting seeing the first gene editing drug, Casgevy, which is developed by CRISPR Therapeutics and Vertex, getting FDA approval earlier this year. Can you just reflect on what were some of the key moments, on the path to approval, and where do you see the field going next after sickle cell disease?
It was very exciting to see, CRISPR Therapeutics getting sort of advanced, especially through the clinical testing and even becoming approved therapeutics. So last December, the very first CRISPR therapeutic has been approved by both the UK and also the US FDA, and that's for treating sickle cell disease. And so I think a number of patients have now received the therapeutic, and exactly as predicted, these are single treatments that result in very, very long-lasting therapeutic effect. So this really proved the idea that it is possible to treat diseases by correcting the underlying genetic mutation. So going forward, there are 5,000 genetic diseases, and then there are other diseases that we can treat by changing the master regulators. So scientists and also drug developers are working to advance CRISPR-based technologies to be able to treat all those other diseases. Sickle cell is a disease that scientists treat by changing bone marrow cells in the body. And so the other diseases affect other organs. Some affect the liver, some affect the brain, and others the eye and other organs in the body. So in order to get CRISPR to work well for these other diseases, we also need to then advance delivery technologies so that we can very efficiently target CRISPR technology into those cells. The technology for accessing bone marrow cells works pretty robustly, and so it's shown to be efficacious for sickle cell treatment. And then the technologies for delivering to the liver is also quite advanced. But for other organs in the body, we still lack efficient delivery technologies. And so that is one area that scientists are still needing to make a lot more progress on.
Yeah, maybe on the theme of delivery, what are some of the hurdles that we need to tackle as a field to increase the impact of these next generation medicines like cell and gene therapies?
Mm-hmm. So delivery is a major challenge for getting really any kind of drug into the body. When it comes to gene editing or gene therapy, delivery is particularly challenging because the CRISPR molecules or other gene therapy molecules, they're quite large. And so it's hard to get large things into cells, which in themselves are quite small. So what people are working on now is that they're using a variety of different approaches, some by borrowing viruses or viral mechanisms to be able to package and deliver therapeutic genetic material into cells. And the others are more based on chemistry, where people are developing lipids, so basically droplets of fatty oil-like material that can sort of wrap around the genetic payload and be able to deliver that into cells. And then people are also working on more physical methods. For example, using ultrasound to transiently disrupt tissue and cells to get to make them more permeable so that things can diffuse in. And so I think it will be really important to explore actively and also broadly all of these different approaches so that we can have a nice toolbox of delivery methods. We're probably not going to solve the delivery problem with just a single universal technology. It's more likely that we need different things for different tissues, and so by borrowing what works for viruses to get into certain tissues and maybe even combining it with other methodologies like the chemistry-based methods or physical-based methods, I think we're going to be able to get access to more tissues.
Great. And then thinking more broadly, any ideas on how to strengthen the ecosystem so that we can enable new biological discoveries to be made and also to get to the clinic cheaper and faster compared to how things are currently being done?
I think there are many things that we can do to optimize and enhance the ecosystem. I think first of all, we have to start from the ground up, and that means we need to nurture. So I think one of the key things, and especially the things that the Center for Excellent Education through RSI has been doing, is to provide kids in school with opportunities in research, and by giving them exposure early on, it can really play an important role in developing their interest and nurturing their career goals to remain in science and to be able to contribute to science. So I think that is really important. So I think as much as we can nationwide, get kids access to research programs and science-related education programs as early as possible could make a huge difference. Of course, the payoff will take some time. It will take a generation's time to really see that investment paying off, but it is something that will really fundamentally advance biomedicine and probably many other fields of science. And then then simultaneously, I think there are things that are important to advance on the regulatory front, because as we develop these genetic-based medicine, and especially as we go after genetic diseases, many of these diseases affect small populations, and sometimes they could have different mutations in the same gene, but still there are different mutations. And so as we develop these technologies that can go and provide single treatment cures for disease, it's very important to think about how do we accelerate the regulatory process or simplify it so that we can, it will be impractical to require phase 1, phase 2, phase 3 clinical testing for these small diseases. And so to the extent that we can build up a body of knowledge about safety and efficacy of treatments and use that to simplify some of the regulatory process, will probably also make it help translation of these technologies sort of spread faster.
Yeah, just really adding a comment on that point. I know that rare diseases are individually rare but collectively common, and it's estimated that 25 to 30 million Americans have one rare disease or another, and with these rare diseases so rare, it's just not economically viable for companies to tackle them today. And there's a disconnect to your point, where it with CRISPR, it is making it dramatically faster and easier to make changes to the genome. And so I think there is definitely an opportunity there in order to kind of think of more creatively about gene editing as almost a form of genetic surgery, where each surgeon doesn't have to get approval for every new procedure they do, because each individual's body is different. So I think that's a interesting theme that won't turn around immediately, but it's a really important one to really increase the impact. I wanted to kind of jump back to your earlier point, on education, maybe just expanding on that point. Any thoughts on how we can, in a way, expand the base of, science education? For example, is it possible to train folks, from community colleges or trade schools into lab work, so that people who don't come up from a conventional 4-year college program in chemistry or biology can pick up lab work and contribute at the bench?
I think you're absolutely right. We can certainly provide education and training for a lot of people through non-traditional through educational programs that are different from traditional 4-year college education. So certainly there could be very focused biotech programs or other types of programs in information technology or energy or other aspects to be able to get them directly and early on involved in these types of more technical industries. I began to work in the lab when I was a high school student. So when I was 16 years old, I began to work in the laboratory. And certainly I have not had a 4-year college education, but with the right mentorship and training, I was able to learn how to practice certain techniques and procedures in the laboratory. And that made me, that helped me to become able to do experiments despite not having yet gone to college. And so I think a lot of those types of education can be sort of provided for people who wanted to get into biotech or other technical industries early on. But the other thing I think is important is we also need to sort of provide more education for teachers at the earlier stages of education. Elementary school teachers, middle school, high school teachers, providing them with more training in science and research will probably allow them to better sort of share that experience and instill excitement in their students about science and technical disciplines. So I think we'll have to kind of upgrade and enhance the entire ecosystem, from the early students to the teachers and mentors, all the way to providing new types of educational programs for helping them enter the technical industries.
Great. And maybe just a last question on my side before we shift over to some questions from the audience. So earlier this year, the CEO of NVIDIA, Jensen Huang, commented that biology has the opportunity to be engineering, not science. So this is kind of echoing your point, on the analogy of CRISPR enabling, an app store kind of thing. But thinking about, again, where we are currently as a field, how far along are we really in shifting the drug development, arena and capabilities from a so-called hardware problem to a software problem?
I think a lot of that transformation is happening now, and a lot of more transformation is going to happen in the decade ahead. The advances in technology and the accumulation of knowledge in biology is what's turning biology from science into more of an engineering discipline. With the sequencing of the human genome and with more powerful techniques like gene editing or spatial transcriptomics or nowadays with artificial intelligence assisted data analysis and experimental design.
Great, thank you. So maybe shifting over to the questions from the audience, the first one is from Sembian, asking about a specific disease, interested in kind of getting a sense for how long will it take realistically for there to be clinical therapy, gene editing clinical therapies for a rare disease like DMD, Duchenne's muscular dystrophy.
There is progress being made, but it will probably still take some more time for us to really be able to see sort of the disease being fully cured.
Now, maybe taking a step back from specific diseases, there's some questions around kind of the broader ecosystem. So one from an anonymous attendee, how does healthy competition and the need for speed to market, for example, with patents and publications, how does competition and speed help drive innovation and ultimately benefit the public?
That's a great question. I think these are all important factors to a healthy and robust sort of economic system. So people there are certain incentives in the medical field, both from treating patients and improving patients' lives, but also economic incentives for the drug developers. And those incentives together, they motivate people to be able to move faster and to be able to ultimately deliver the medicine sort of as quickly as possible. But one thing that is important is at the same time, the regulatory system and also just in general, the process of developing drugs is that we have to make sure that we are advancing things that are safe and we're not taking unnecessary risks. And so I think it's an important balance to get right, which is both supporting rapid progress, but at the same time, not going too fast without the necessary regulatory and oversight to make sure that things are being done in a responsible and also ethical way.
Yeah, you mentioned ethics. Maybe just expanding on that, there are a couple questions coming in, around, do you think that, CRISPR or other gene editing technologies will have ethical implications? And, how do we think about, gene editing for potentially enhancing physical characteristics, and what are your thoughts on those topics?
Yeah, I think these are important questions and important things to discuss, as a society altogether. Suppose we can use gene editing to change human characteristics and traits. Should we go and do that? Right now, we're still in the phase where for most things that people want to do, probably actually 99.9% of things that people want to do in the form of enhancement, we actually cannot do that yet. If you wanted to make someone taller or change IQ by 30 points or something like that, we really don't know enough about biology, and it's unclear how soon we're going to know that. Biology might actually be much more complicated than we can imagine, where we're not going to have the answer to doing those things anytime soon. And so for those things, we're still in the realm of we cannot do that yet. We just don't have the ability. But ultimately, at some time in the maybe somewhat distant future, we will have that ability, and then it will become important to kind of decide, should we do that. I think as a society, for things that can provide benefits to human health and also human society, I think ultimately people will probably opt to do those things because you can see with things that try to improve people's health or cosmetic surgeries or even things that the parents do to help their kids get into college. You can see that they really will do whatever they can to try to provide an advantage for advancement and also survival. So I think ultimately a lot of these things will probably happen, but it's really important to think about what is the most ethical way to allow those things to happen in society. I think we'll still have some time to work through those hard questions. These are questions that have been around for a long time, and I think people are continuing to grapple with it. The recent development of gene editing over the past 10 years, in some ways, has sort of reignited a lot of the discussion around these ethical topics. So I think that's a really good thing, and we need to really engage as many aspects of society to talk about it as we can. These are not simple questions.
Yeah, thanks. And maybe shifting gears, there's a question from Ezekiel about agriculture. Can you describe, what kind of impact CRISPR has had, on agriculture, biotech? And there also are some analogous questions on are there some boundaries on whether there should be limits to how CRISPR should be used, but what are some thoughts on appropriate uses of CRISPR in ag?
CRISPR is being used to advance agricultural science and also agricultural development quite a bit. Scientists are using it to understand genes and their regulatory function in supporting crop yield or drought resistance or pest resistance or any other number of things. And so those are very important things that people are using CRISPR to learn. But what they learn from those experiments can also result in translatable enhancements to crops and other agricultural products. And so that is also really good. We do face with, as a society, food security problems, and also the changing climate will make crop yield and also just general food security much less predictable and constant. And so using CRISPR to be able to accelerate the solution to some of these problems, I think is going to be really important, and it's something that probably we need to do even more. One thing that is worth mentioning is that using CRISPR, CRISPR is different from GMO. Rather than introducing foreign genetic material into a plant, what CRISPR does is that it's simply making mutations that you would be able to get through traditional crossing and breeding. So the difference is that CRISPR can achieve those outcomes a lot faster than crossing and breeding, because you don't have to wait for multiple generations of the crop to achieve the outcome. You can combine all those genetic traits together in fewer steps. And that acceleration is really important and also helpful for developing progress in the agricultural field.
Thanks. Maybe shifting back to the topic of education, there's a question from an anonymous attendee who's interested in CRISPR and is from a rural area and is wondering if you have any advice on how to get hands-on experience, whether it's online programs or even just finding a mentor. Any thoughts on that?
I think there are a few things that are sort of accessible. So online, there are a lot of content, both things in terms of research articles, but also a lot of podcasts and webcasts and even video sort of presentations on the CRISPR field. So I think accessing some of those material can provide more of a theoretical education in the gene editing and CRISPR field. In terms of getting hands-on access, I think it's really important to then try to get access to a educational resource, whether it's a laboratory or some type of courses where they are teaching these techniques. So I think those are available. Probably there are probably researchers working with CRISPR at most universities in the country. So I will try to find one of the local universities, maybe reach out to some of the laboratories to see if you can volunteer in those labs. A lot of laboratories have volunteers as young as high school students, and so that might be one way to get some firsthand experience.
Yeah, and we're rapidly coming to the end of the hour. So this will be the last question from the audience. This is from Owen, wondering what are the key qualities and skills that you believe are essential for success in a biomedical research career, and what strategies do you recommend for staying current with the rapidly evolving scientific literature?
These are really good questions. I think what is really important in biology is to kind of develop to nurture your curiosity, sort of have curiosity for different problems and constantly ask questions, because biology in some ways is based on a scientific process. You try to generate hypotheses and then be able to develop ways to test those hypotheses. And so the more questions you ask, the better you'll become at generating hypotheses. At the same time, I think developing a multidisciplinary skill set will be important. Rather than only learning biological techniques, learning coding and learning computational techniques and learning chemistry and physics could help you to bring additional insights that a traditionally biologically trained scientists may not think of. And so these kinds of curiosity and also more exposure beyond biology is probably going to be really helpful. In terms of staying current with rapidly evolving scientific literature, I think there's so much being generated, but I think this is also where probably trying to stay on top of some of the major publication and news outlets would be helpful. Reading magazines like Science and Nature and also reading review papers for specific fields will probably help provide a broad perspective, especially for someone who is trying to get into a new area. Finding some of the authoritative reviews that are published in major review journals can give you that overview, and then based on the overview, you can then zero in on specific topics within a specific problem.
Fantastic. Well, just wrapping up, I've really enjoyed the discussion here today. I'm leaving feeling inspired. And we clearly see rapid progress here with CRISPR. I think we also can think of CRISPR as a trailblazer for new platform technologies. We didn't have a chance to touch on Feng's latest research. A lot of really exciting stuff that you're doing. There's also as we all know, a lot of exciting work that's happening with generative AI, and it's being applied to designing proteins and other therapeutic molecules. Those we could spend an hour each, on those topics alone. But I'm convinced that if we fast forward 10 to 20 years and we look back onto the state of the art today, we'll think that things were so primitive back then, just by the pace of progress that we're making. So I think in today's conversation, it's broader than CRISPR or gene editing. It's really about how to create the environment and the ecosystem that can enable new technologies to come into existence, going from 0 to 1, and also to have a positive impact broadly, so-called, going from 1 to 100. And I think that with technological change, it's inevitable that there will be winners and losers. And I think it's safe to predict that the best minds and hands in life sciences will be among the winners. So on that note, I think this is a great place to wrap up today's discussion. I'd like to thank Feng for your thoughts and for your time, and also to Mrs. D and the CEE for hosting today's webinar to help advise the Congressional STEM Education Caucus. So thank you all.
Thank you, everyone.